us today for our first of what hopefully will be many webinars from the Prostate Cancer Foundation, which our CEO, Jonathan Simons, has renamed the Prevent Coronavirus Fatality Foundation. So we have noticed a huge outpouring of research from cancer scientists all over the world in the midst of a crisis when people can't effectively work in their laboratories on cancer biology that they've been trained their whole career to do. They have rapidly responded to the crisis and are working on coronavirology, coronavirus pathology, epidemiology, and everything you can imagine. My partner in science, Dr. Miyahira, and I have reached out to over 120 of our young investigators and have found, I don't know what the current count is, Andrea, but it's somewhere around 50 or 60 people have dedicated day and night work to coronavirology and pathology and epi and everything else you can possibly imagine. So we're gonna go through that today. We've chosen some expert speakers. This is going to be a rapid fire event that will start with an introduction on TMPRSS2 because that's our focus today. TMPRSS2 biology, pathobiology, and epidemiology. We have Christina Jameson from UC San Diego who will provide an introduction. And then there will be scholarly talks for approximately 10 minutes each. Carlos Corden-Cardo, who's the chair of pathology at Mount Sinai Hospital in New York City at the Icahn School of Medicine, will be first, followed by Dr. Arul Chennaiyan, a professor of pathology at the Institute of Translational Pathology at the University of Michigan. And congratulations again, Dr. Chennaiyan, on your rising to membership in the National Academy of Science. We're just all thrilled about that. Followed by that will be Nima Sharifi, who's a professor of medicine and one of the world's leading androgen pharmacologists and biologists focusing on prostate cancer from the Cleveland Clinic in Cleveland, Ohio. And Andrea Alamonte, who's a professor of oncology research at the University of Padova in Bellinzona, Italy, who recently published a very provocative study on the epidemiology of COVID infection in patients undergoing treatment of prostate cancer with androgen deprivation therapy. So it's very much a TMPRSS2 meeting today. The next webinar that we have in a few weeks will be on emerging clinical trials that are starting and will start all over the world, asking important questions about new treatments focused on TMPRSS2. And it's now with my great pleasure to introduce Dr. Jameson from UC San Diego, who will give an overview of TMPRSS2 biology and biochemistry. Dr. Jameson?
Great. Thank you, Howard, and thank you very much for hosting this and organizing this incredibly exciting and important topic. So I'm going to ask the question, why TMPRSS2? And so TMPRSS2 is a transmembrane serine protease. It's one of the very few trypsin-like proteases that was known at the time to be expressed in human respiratory tract when it was looked at for being involved in viral infection. It was shown to be the protease which could activate a viral infection of human influenza virus in 2006 and also SARS-1 now. In prostate cancer, of course, we're very familiar with it because it's part of the TMPRSS2 ERG chromosome translocation, which is found in approximately 50% of primary prostate cancers and 30% of metastases. So we've studied this protein and gene intensively, we meaning the leaders in the field who will follow, and we know that it's androgen-regulated and highly expressed in prostate. So in terms of SARS-CoV infection, what has TMPRSS2 been found to do? So recently there was a paper published by Hoffman from the group of Pullman, who's also involved in these studies, showing that SARS-CoV-2 entry depends on ACE2, but also TMPRSS2. And they also showed that there's a protease. And so what is it doing? So the coronavirus actually binds to the cell by ACE2, and we kind of knew that before, and its attachment is mediated by the N-terminal receptor binding domain. But its viral entry requires exposure of the fusion component, which requires proteolytic cleavage. And this is carried out by TMPRSS2. In looking at the cleavage sites, they noted that the SARS-2 S protein had conserved cleavage sites. And so they tested to see if SARS-2 was using the same ACE2 TMPRSS2 viral entry mechanism. And they showed using a pseudotype or fake virus, where you can use a construct that has any viral coat protein that you want on it for a single cell infection, and use luciferase to follow viral entry. And they tested viral entry in a panel of human and animal cell lines from various tissues, and showed that the pattern was the same for SARS-2 and SARS-2 for SARS and SARS-2 entries. So they show then in the paper that the entry is actually mediated for both SARS and SARS-2 by ACE2 and TMPRSS2. They show it by also using authentic virus in a lung cell line, and they use an inhibitor of TMPRSS2 serine protease to show that that inhibitor could actually inhibit viral entry here of the authentic virus, and then here in primary human cells using the SARS virus VSV pseudotypes. So the highlights of this paper were that they showed that SARS-CoV-2 uses the same ACE2 to bind, for the S protein to bind to the host cell, and then the TMPRSS2 proteolytically cleaves, exposes the fusion peptide, and allows viral fusion. This process can be inhibited by chemostat mesylate, which is clinically approved in Japan and might be a treatment option. Interestingly, they found that antibodies that were from patients who had recovered from SARS-1 could give some cross-protection against SARS-CoV-2 using this pseudotype virus. So where are these two genes co-expressed? And in looking at SARS-CoV-2 tissue tropism, in other words, what organs might be very sensitive to SARS-CoV-2 infection. And so in this paper from Sungnak et al. In Nature Medicine, they looked at multiple single-cell RNA-seq data sets across different tissues. And if we zoom in on the respiratory tree, we can look at ACE2, which is represented as a dot, a percentage of cells in that sample that express the ACE2. Here, second column is TMPRSS2, and the third column shows the co-expression. You can see nasal cavity, ciliated and secretory cells, but if we go down here to prostate, you can actually see that there was co-expression in normal prostate cells in these Hillock cells. So what about prostate cancer? So we will hear a lot more about that in the next talks. I'm very excited about that. We looked in our own data where we had RNA-seq analysis of castrate-resistant prostate cancer bone metastasis 3D organoids. And we had looked at TMPRSS2 and could see expression of TMPRSS2 as expected, but we also found expression of ACE2, experiment 1, experiment 2, albeit at very low levels. Interestingly, depriving the organoids of androgens or treating them with enzalutamide led to decreased expression of not only TMPRSS2, but also ACE2. So these preliminary data indicate that prostate cancer may be a target tissue of SARS-CoV-2, and that anti-androgens may reduce the viral entry by reducing the expression of the host factors. So thanks.
Thank you very much, Dr. Jameson. Before we head into our major talks, following that very nice overview of TMPRSS2, I just have a couple of housekeeping points to make. At the bottom of your Zoom screen, you can see a Q&A button. Please ask lots of questions. We can pretend it's a Coffee Holden meeting for those of you that have been to that PCF event. You can ask questions and you can vote thumbs up on other people's questions. And we will actually use the results of that polling to decide which questions will be addressed in the Q&A session after the talks are complete. Secondly, I'm going to turn the mic over to Dr. Miyahira, who will tell you about a poll that we've made, that she has made, that will be a very interesting result on who's doing what these days.
Yes, thanks. Everyone should see a poll pop up in their screen just now. It's just asking whether or not you have pivoted your research to COVID-19. It's multiple choice, so if everyone on the webinar could just answer, I'll give about another maybe 30 seconds for everyone to fill in their answer. I'll just leave it open for another 30 seconds or so, but we can move forward.
If you can't tell, we've never done this before. This is our first shot, so thank you for your patience. Our first speaker will be Dr. Corden Cardo from New York, who will talk to us about some very provocative, important, and interesting phenotypes that he's discovered in COVID-19 patients. Carlos?
Yes, thank you very much. Just want to share with you our results, and wanted to divide the talk essentially in two specific themes. The first one, I'm going to share with you how we have been testing and some interesting findings from our autopsies, and then I'm going to turn into phenotype expression as it relates of terms and androgen receptor and ACE2 in different age and different genders, and some interesting results that we have observed on that as well. So we produce an end-to-end pathology solution to look at the disease from the point of view of diagnosis, not only qualitative, but also with viral loads and phylogenetics. We have tested serologically and also trying to identify some potential targets for therapy. We have a therapy for this disease, which is our own antibodies, and also for some of the symptomatology. And we have conducted over 80 autopsies, which gave us a great deal of interesting insights that I would like to share with you. We have done over 35,000 diagnostics, and we have done over 30,000 serologic tests. Interestingly enough, this disease, it's a disease that we didn't see before. It's a new disease that has two different targets. One is the target that we have been discussing, essentially, secretory epithelial cells. We have looked at all of the organs in complete autopsies, including not only the technologies that they are conventional, such as H&E, but also electron microscope analysis, as well as a series of immunohistochemistry and in situ hybridization. We have seen the typical damage in the lung with very abnormal atypical cells, which are the cells that engulf and they are attacked by the virus, shown also in electron microscopy with immunohistochemistry. And in situ, we have seen presence of the viruses in these cells. But very interesting, we have seen another disease, a disease that happens inside the blood vessels, that is produced by a hypercoagulable syndrome and the presence of microthrombi. This has been seen not only in the lung, but also in other organs. In the heart, there is very little inflammation, but again, thrombi. And in the bone marrow, as well as in the spleen, we have seen a very interesting development in the context of macrophages engulfing cells, the typical phagocytosis, which is also related in the context of the macrophage activating syndrome, something that we can discuss later, which can be part of the cytokine storms that we are seeing. And interesting enough, in many organs, including the kidney, we have seen the presence of ACE and TMPRSS2 in the epithelial cells, but not in the endothelium. The same in specific other organs, including the brain, in which we have seen the presence of ACE2 in the endothelial cell, in the capillary network, as well as in the venules and in the blood vessels, producing these thromboembolisms and microthrombosis, that produces several diseases in these individuals. With this in mind, and also knowing the natural history of TERPs and how it's related to prostate cancer and to ACE, we wanted to find out what happens in the context of specific questions, like, for example, why children and young individuals don't get as sick. One of the things that we did was an analysis of the androgen receptor with the right controls, TMPRSS2 and ACE2 in different organs. As you can see, the controls work well. In the prostate, we have in the epithelium both TMPRSS2 as well as ACE, as it happens in the, testes. But very interesting results were very different in some other organs. For example, the androgen receptor in the bronchial epithelium, it's quite expressed in young adolescent males and in old males, but not in females, both young and old, and in young females as well. The same, it's with specific expression patterns with ACE and TMPRSS2, as I'm going to share with you. We look at some other epithelia that can be also in the oropharynx and part of the syndrome. And the same, we have seen that, this is very sensitive, that in the tonsillar epithelium of both young and old males, the androgen receptor is expressed. We have, I'll share with you also some other phenotypes, but we have seen no expression in older female or younger females. So more specifically in the respiratory epithelium, we have seen the presence, as I mentioned, of the androgen receptor in some cells. ACE is quite expressed, TMPRSS is quite expressed, but it's also very much expressed in the endothelial cells. Another area that I think that we need to pay attention, because this can be an important target in the context of the disease. Similarly, in the small intestine or in other tissues, we have seen the presence and the co-expression of both ACE and the TMPRSS. And we have seen also in the heart and in the brain this expression of ACE2 that may be related to some of the pathogenesis that we are seeing in the disease. So, summary from these findings: this is a different disease. This is a disease that not only affects the epithelial cells, but also the blood vessels. Studies that we have been conducting points out that we need to pay attention to some of these other microanatomical localizations. There is a very differential expression in age, maybe being related to the transcriptional activity of the androgen receptor upon TMPRSS and the association with ACE2. We need more data and we need to look further into that. And the fact that put it together, we are not seeing as much inflammation as we thought we were going to see in the context of lymphocytic inflammation, but actually we see quite a bit of macrophage activity and what we can describe as a macrophage activating syndrome. Howard.
Thank you very much for that. Andrea Miyahiro, would you like to put up the poll? Do you have the poll summarized? There you go. So 40% are not conducting research, 32% have begun, and 19% are planning on it. So just about everybody's thinking about this virus and this infection and this disease. So thank you, Andrea. Our next speaker from the University of Michigan is Dr. Arul Chanayan. Arul, we look forward to seeing what's going on in your labs in Ann Arbor. You're on mute, Arul.
Good. Can you hear me now? Hello? Yes?
Now we can hear you.
I just started here without just speaking to my screen here for a bit. But no, thanks to Christina for the nice introduction, because I think I can move through some of my slides very rapidly, as well as some of the experiments that we've carried out nicely overlap what Carlos presented as well. So it's nice to see some corroboration there as well. So I will sort of move through this pretty quickly here. I won't go through the background here because I think Christina covered this quite nicely. But then sort of jump right into the general hypothesis here is, can we think about some of these FDA-approved therapies that work in prostate cancer that suppress TMPRSS2 expression in terms of AR signaling, and can that potentially be repurposed to block coronavirus infection and replication? So that's, I think, one of the hypotheses that we're talking about today. But there are a number of open questions when we think about this. One question being, let me see if I can get my pointer working here as well. Or a few of the open questions really here are, do TMPRSS2 or ACE2, which is actually on the X chromosome, do the levels of those proteins explain the gender disparate effects of COVID-19? Is TMPRSS2 indeed regulated in the lung lineage, regulated in the prostate lineage, but outside of the prostate, it's unclear whether TMPRSS2 utilizes the same regulatory machinery being driven by AR and potentially other steroid hormone receptors. And then I think the other question, the last question that at least I'd like to propose is whether direct inhibition of TMPRSS2 could work, or potentially indirect inhibition of TMPRSS2 through transcriptional targeting could potentially work in terms of having a therapeutic effect in COVID-19 disease. So just to begin, I just wanted to share with you a SARS-CoV-2 bioassay and screening approach that we're working with Johnny Sexton here at the University of Michigan, who has a BSL-3 facility. And this has been a powerful resource potentially to screen new drugs for their activity against SARS-CoV-2. And so what we have here basically is in blue is, of course, nuclei. In green are lipid droplets. And it's known that SARS-CoV-2 affects lipid metabolism quite significantly. And then in red, we have the SARS-CoV-2 nucleocapsid protein. And so this is the negative control here, and you see nice infection. However, when you hit these cells with nanomolar levels and even 50 nanomolar levels of remdesivir, you see marked inhibition of viral replication. And you can actually see abortive replication here with the various arrows pointing to these red dots. So you can see that certainly remdesivir is quite powerful in inhibiting infection and replication of SARS-CoV-2. Now as sort of a control, and I know that this has already been shown, but using this particular high throughput screening assay, I'm showing you some of the data here with the TMPRSS2 inhibitor, Camostat, which is actually FDA approved to treat pancreatitis in Japan, but now is being repositioned in a trial in Denmark, looking at COVID-19 patients. You can see the Camostat inhibitor, again, markedly inhibiting CoV-2 replication and binding and so forth. And here is the dose curve of that. So now getting back to the hypothesis of AR regulation of TMPRSS2, here I'm just showing you data in prostate, and certainly many groups have shown this in different contexts, that certainly in the prostate lineage, by ChIP-seq analysis, AR binds to a particular enhancer in TMPRSS2, and this is shown here with synthetic androgen, and that this can be blocked by enzalutamide. We've shown that basically using synthetic androgen, you can induce TMPRSS2 expression, as well as block its expression with enzalutamide and anti-androgen. So certainly this is quite well established, I would say, in the prostate lineage. It would be nice if this was analogous in the lung lineage as well. So now beginning to explore that question in various cell lines outside of the prostate lineage, looking at lung and colon lineages. And we've actually screened a number of cell lines, and these are, of course, lineages that are affected by SARS-CoV-2. So we've identified a number of cell lines here. I'm just going to show you data from a lung adenocarcinoma cell line that has high levels of AR, TMPRSS2, and ACE2. And what we found in this particular cell line was that TMPRSS2 was not regulated by androgens with either enzalutamide or an AR degrader. You see equivalent levels of TMPRSS2. Similarly, ACE2 is not regulated, nor was AR in a lung lineage cancer cell line. Likewise, when we look at primary lung alveolar epithelial cells, we again did not see regulation of TMPRSS2 using darolutamide, an AR degrader, or we didn't see regulation of ACE2 in primary pulmonary alveolar epithelial cells. And this is just another cell line just to show that the lung cell line wasn't a peculiarity in a colon cell line, again, that has AR, TMPRSS2, and ACE2. These are not regulated by dihydroxytestosterone and cannot be blocked by enzalutamide. So again, these are cell lines outside of the prostate lineage. Now to take a deeper look into the actual cell types that are that express these key proteins, that being AR, TMPRSS2, as well as other steroid hormone receptors. We took advantage of, or we analyzed publicly available single cell sequencing data. We actually looked at upwards of 8 different single cell sequencing data sets, which included both human as well as mouse lung single cell sequencing data, so that we can look at the resolution at the single cell level. And when you focus in on the key players here, TMPRSS2, AR, and so forth, and this is relatively consistent across data sets, but what I can say is that in general, single cell RNA sequencing data can be quite noisy, so you certainly have to look across data sets. But we'll just focus on this data set, which I think is relatively representative. You can see high levels of TMPRSS2 in alveolar epithelial cells, both type 1 pneumocytes as well as type 2 pneumocytes. You see lower expression of AR and reasonable levels of ACE2 in alveolar epithelial cells. When we look at bronchial epithelial cells shown here, we're seeing again quite reasonable levels of both AR, TMPRSS2, and ACE2. So all of the players are in the right cell types, that being bronchial epithelial cells, as well as in alveolar epithelial cells. Although the balance of the levels of these proteins or transcripts might be different. Now jumping into in situ analyses, looking at the actual tissue specimens, and certainly I think we initially tried to explore this using tissue extracts, but we found considerable heterogeneity based on the admixture of cells potentially that are in a tissue extract, meaning the balance of bronchial epithelial cells and alveolar epithelial cells. So we've really focused our analyses on single cell sequencing data as well as in situ type analyses. Here I'm showing you immunohistochemistry data that actually Carlos showed as well, that basically we see very high levels of AR protein in human lungs, in male human lungs versus female, and this is shown here. And this is in the bronchial epithelia, as well as in the alveolar epithelia and alveolar stromal cells. We see very high levels of AR protein in males versus females. And this is a quantitation of that data. Again, very high levels in males versus females in lung, both bronchial as well as alveolar epithelia. When we try to translate these results into the mouse preclinical systems, we again see very high levels of AR protein in male mice versus female mice, so that's nice. And then interestingly, to try to emulate anti-androgen therapies, we took advantage of our castrated mice and compared intact mice versus castrated mice, and we saw a marked decrease in AR levels upon castration. So this is good because this suggests that you can at least extinguish AR expression in the lung. Now shifting gears to TMPRSS2, we have been working up various antibodies against TMPRSS2. Many of the commercially available antibodies are not, do not validate quite well, but we believe that we've identified better TMPRSS2 antibodies. But here I'm showing you in situ data for TMPRSS2. And the little brown dots basically represent TMPRSS2 transcript levels. And you can see that we're not actually seeing a difference between females on the top here compared to males on the bottom. And this is quantitated here in terms of bronchial TMPRSS2 expression. However, when we looked at alveolar TMPRSS2 transcript expression, we're starting to see a trend of increased level of TMPRSS2 transcript expression in male lungs versus female lungs in the alveolar epithelia. This hasn't reached significance yet, but we only have 5 in each category of 5 males and 5 females. So I think as we increase our numbers, we might see a significant difference. So I would just say it's trending in the right direction. And then finally, looking at TMPRSS2 and ACE2 in the murine systems, what we found was that TMPRSS2 in alveolar TMPRSS2 transcripts were again decreased upon castration, as well as we saw a difference between male and female in terms of TMPRSS2 levels in alveolar TMPRSS2. Likewise, with bronchial ACE2, we saw actually a marked decrease in ACE2 transcript levels in castrated males relative to intact males, and females relative to males. And this was actually, this nicely connects with some of the data that Christina presented earlier, except in an animal model of castration, we see a marked decrease of ACE2 levels. So this would again suggest that an anti-androgen type therapy could have potential in not only suppressing TMPRSS2, but also ACE2. So finally, just some take-home points here. And in blue, I highlighted those points that might be consistent with the hypothesis that blocking AR signaling and TMPRSS2 could work in treating COVID-19 disease. And in red, I'm just highlighting observations that we have that aren't necessarily consistent with that hypothesis, or essentially the null hypothesis. So I would say the TMPRSS2 is certainly an attractive target in the treatment of coronaviruses, as well as influenza viruses. Just to mention that influenza viruses also use TMPRSS2 to enter the host cells. The cell line systems, the lung lineage cell line systems, both primary as well as cancer cells, TMPRSS2 does not appear to be under regulation of AR. In terms of single cell RNA sequencing data, this certainly suggests that all of the right players are in bronchial epithelial cells as well as in alveolar epithelial cells. In terms of AR levels, in both human and mouse, we saw much higher levels in males versus females. In mice that were intact versus castrated, we saw much more in intact, and it markedly decreased upon castration. So this is consistent with the hypothesis. In the mouse and human bronchial TMPRSS2 levels were about the same, so not much of a difference there. And the bronchial epithelia do make up a large fraction of TMPRSS2 expression in the lung, but we're not seeing a difference between males and females. However, in the alveolar TMPRSS2, we're starting to see a signal both in human and mouse systems that males might have higher levels than females. When we look at bronchial as well as alveolar TMPRSS2 in terms of our murine models, again, upon castration, we see a decrease in TMPRSS2. And probably most interestingly, in terms of the bronchial ACE2 levels, we see much higher levels in males relative to females. And this level of ACE2 is markedly decreased upon castration. And so that summarizes the data here, and just to acknowledge the various individuals in the group that contributed to a number of the bioinformatic analyses that I showed here, as well as the lab experiments and the literature review, as well as our collaborators.
Thank you, Arul. Splendid work. I just remind people this was done during a period of time when the labs were essentially closed to cancer research everywhere. So thank you to your investigators and for your leadership to get these important laboratory findings in the bank. Our next speaker is Dr. Nima Sharifi from the Cleveland Clinic, who's also managed to publish a heroic study. So Dr. Sharifi, we look forward to hearing your thoughts.
Okay, can you hear me and see the screen?
Yes, if you could just change your display settings, we'll get it all on one screen, will be great.
How do I do this here?
I think Andrea Miyahira knows how to resolve.
Yeah, maybe you can advise me on this, Andrea.
Just share your screen again, Nima, and then at the top of the PowerPoint, you could choose swap display settings.
Sorry, I don't see.
It's okay, put yourself in presentation mode.
How's that?
And now it's display settings, you can swap.
There we go. How's that?
That's perfect, thank you.
Okay, sorry about that. So I want to start by thanking Howard and Andrea and the other panelists for the invitation to participate in this knowledge exchange on COVID-19. So as was outlined quite nicely, there are at least two possible androgen-driven mechanisms that can underlie the sex discordance in COVID-19 outcomes, which is specifically that outcomes in mortality tend to be worse in males compared with females. The first possibility relates to androgen-driven regulation of TMPRSS2 in pulmonary tissues susceptible to infection, and possibly effectively raising the levels of that co-receptor that's required for viral infection. The second possibility has to do with androgen-driven immune regulation. The existing data include observations that testosterone deprivation in men may be immune stimulatory, and that a number of inflammatory processes tend to be worse in women compared with men. So one example is in asthma, where asthma tends to be worse in boys compared with girls. And then after puberty, with the onset and rising of gonadal testosterone, things tend to get better in men and worse in women. So together, this may speak to an immune suppressive effect of androgens. So to address the TMPRSS2 hypothesis, we looked at transcript expression in seven different data sets, comparing male versus female lung tissues. So these are together widely representative data sets that come from lung parenchyma, large airway epithelia, small airway epithelia, bronchial epithelia, and then also from smokers and non-smokers, which I think is quite important. And we didn't see any elevation of TMPRSS2 transcript expression in human male pulmonary tissues. We also did look at AR transcript levels and similarly found no elevation in males compared with females. So this is an analysis done by Jeff McManus. What we did find, and what I'm not showing you here, but it's in the bioRxiv paper, is that TMPRSS2 expression tends to be elevated in current smokers, but not former smokers. So we next wanted to test whether TMPRSS2 expression could be suppressed with the AR antagonist enzalutamide in a mouse study. So this is work done by Mehdi Barachian, who's a postdoc in our group. And what he did is he treated intact male mice with 11 days of enzalutamide compared with chow. So about 10 or 11 days, because this is about when we see the onset of some results, some regression with xenograft studies. And so we see no difference in TMPRSS2 transcript. Again, this is, so the way this was done is that we basically, after we sacked the mice, isolated an entire mouse lobe and then developed generated RNA or protein. So at the level of the whole lobe, we see no difference in expression between intact males and females, and then also no difference in transcript expression with enzalutamide treatment. At the transcript level as well, for AR, we see no difference between males and females. There was a slight, significant but modest increase in AR transcript levels with enzalutamide treatment. So at the protein level, we tested 2 different antibodies. Again, this is the entire mouse lung lobe. There may be a slight difference in TMPRSS2 with one antibody, but certainly not with a second antibody. So looking at the entire lobe, there was no gross difference. And then with enzalutamide treatment, we didn't appreciate any suppression of TMPRSS2 protein. So the caveat here obviously is that we didn't look at this at the tissue or single cell level. This is with the entire lobe. The other thing that I'll mention is that similar to what Arul had mentioned, there is a pretty big difference in AR expression in males compared with females here. So there's one more thing I want to just share along the lines of androgen immune regulation, and specifically in that there's not evidence just for immune regulation by gonadal androgens, but also by adrenal androgens. And there's some evidence that this is genetically regulated. So this evidence comes from another pulmonary disease, asthma. And this is a story that we published in PNAS earlier this year. So in low DHEA settings, and this seems to apply at least for inflammatory conditions so far as we can tell in low DHEA settings. When you inherit this adrenal restrictive or slow enzyme, this will limit the conversion of DHEA to potent androgens in peripheral tissues, and you would expect may not induce immune suppression. But with inheritance of this more active or adrenal permissive enzyme, this would enable the generation of potent androgens and may induce immune suppression. So the data here are from 2 pretty good size cohorts of asthmatics. These are multi-institutional NHLBI funded studies with over 300 patients to the left, about 200 patients to the right. What we're seeing here on the y-axis is FEV1% predicted. So it's a measure of lung function, probably the best indicator of lung function for asthmatics if you speak to the pulmonologist. So the higher the better and the lower the worse. And these are the 3 different genotypes: adrenal restrictive, 2 copies of the adrenal restrictive allele here on the left, 2 copies of adrenal permissive or the fast enzyme all the way to the right, heterozygotes in the middle. And the phenotype really seems to come out specifically in low DHEA settings. And in that when for patients who inherit 2 copies of the fast enzyme, they have a lower severity of asthma, specifically when they have low DHEA here and also in the second cohort. So this is really consistent with this effect being mediated by potent androgen-induced immune suppression. So just to say that when we're discussing effects of androgens on the immune system, I think it's important not to forget about adrenal androgens and their potential effect. So I'll stop there. Thank the folks who actually did the work and our collaborators, and thank you for allowing me to participate.
Great, Nima. Thank you. Thank you so much. And thanks to your laboratory staff that, along with you, were sufficiently brave to go to work and get that work done. So our final speaker before we get to questions and answers is Andrea Alamonte, who I know as more of an immunotherapy physician scientist working with Johan de Bono on IL-23 and other important problems. But, you get an email on a Sunday night to learn about a paper coming out. So he's been working with the epidemiologists in his institution in Italy and right on the border with Italy and Switzerland. And he's going to share that data with us today. Thank you, Andrea.
So thank you very much, Howard. Thank you for everyone for this opportunity. So, you know better than me that when this infection hit Italy, we immediately realized that SARS-CoV-2 infected men have a worse clinical outcome than women. And so we immediately put forward this hypothesis, can androgen explain this increased vulnerability of male to develop infection that are severe infection by COVID, the SARS-CoV-2. And this has been shown already by previous speakers, is the rationale behind this, or mean what we thought could be the rationale. So there was a beautiful paper published on Cell by Hoffman that showed that TMPRSS2 is needed for the virus to infect the cell. And we have heard about the possibility that TMPRSS can be regulated by androgen. So we have also data in our laboratory that of course androgen can increase the level of TMPRSS in vitro. However, as reported also by others, we at the beginning were shocked by the fact that androgen deprivation therapy, particularly enzalutamide or castration in mice, didn't decrease also the level of TMPRSS2. So we were also thinking about other possibilities by which androgen could promote or favor the infection by SARS-CoV-2 in male. And there have been a lot of studies back in the 60s and the 70s and later on regarding the possibility that androgen can regulate the immune response. And in particular, my lab worked on cancer immunotherapy and in particular in myeloid-derived suppressor cell, which are neutrophile. Been shown that androgen, for instance, can regulate the number of neutrophils, so more androgen, essentially more neutrophils, and androgen can also regulate the function of these neutrophils. So you may also be aware that there have been publications showing that patients affected by SARS-CoV-2 that have a severe complication have an increased NLR, so more neutrophil and less lymphocytes which are circulating. So we wanted really to go to the clinic and propose to our colleague to do a clinical trial, but they were not satisfied about this evidence. So I tried to think about the possibility to convince the clinician to run clinical trial, and I thought that the only possibility would be to look into a population study. We working in the prostate cancer field know that male are affected by prostate cancer are treated by androgen deprivation therapy. And so thanks to the help of the region of Veneto, which has been one of the most affected regions in Italy by this terrible infection, we were able to study a population of 9,000 SARS-CoV-2 infected male. Between these 4,500 were male, and we further zoom in this population to look at how many of these patients have cancer or non-cancer, and how many of the cancer.
Andrea, we lost your voice. I think we've lost Dr. Alamante. Andrea Miyahira, can you, do you know what's happening?
Let me take a look. I see him, let me see if I can send him a chat. I suspect he dropped off. Do we want to give him a minute to get back on?
Why don't we invite Pete Nelson forward? Dr. Nelson from the Fred Hutch had a question about estrogen versus testosterone in this system. Pete, maybe you could ask that question to our panelists.
You bet. Can you hear me okay?
Perfect.
Yeah, great. So it's primarily for Dr. Cordencardo, but I think any of you can address it with the molecular data. So there's some evidence that TMPRSS2 may be estrogen regulated in certain cells in certain situations, maybe even differentially with ER alpha and ER beta. So my question first to Dr. Cordencardo is, have you done any immunohistochemistry? You've nicely shown for androgen receptor, but is there any evidence that either ER alpha or ER beta may be co-expressed in women, and explain perhaps some of the relationship with infection? And then we could look also at sort of parallels where you've nicely shown androgen perhaps deprivation therapy reduces either incidence rates or aggressiveness of infection, and think about ER antagonists that women may be taking, and look epidemiologically and see if there's a relationship with tamoxifen or raloxifene or some of these. So kind of a long question, but multiple parts, and I think you all have data that could potentially look at the ER effect in women.
So it's a great question, Pete. And as a matter of fact, in Stony Brook in New York, there is already a clinical trial ongoing using estrogen inhibitors, estrogen receptor inhibitors in a subset of these COVID-19 patients. We have plans to do this study together with the group of Kenneth Schroyer there, but we have not initiated because I think that we have no time to think and lots of time to work. I mean, it has been very active here in New York, but that's a great question. But the clinical trial has initiated, it's my understanding, in a subset of patients.
So I think this will be really interesting to address, Pete, when we start interrogating the clinical trials in a couple of weeks, when we get that webinar scheduled, because we'd like to see women treated in these trials as well as men.
Right, so the molecular data, so like Arul and Neiman, did you guys look at ER expression in the different lung cell types or respiratory respiratory cell types?
Hey, Pete, how you doing? We've looked at single cell sequencing data, and I could probably even, I think I've actually shared that earlier, but let me see if I can throw that up here again. We've got time.
While you're doing that, actually, Marco Guitardis also asked about GR. So
yeah, actually, that GR is in the mix too. So, but here you can see basically. So we have AR. Can you see my arrow?
yes.
AR, ER, and GR here. So GR is expressed sort of across the board in terms of number of cell types by single cell analyses, while ER is at relatively low levels, I would say, across most of these data sets, but again, within the range of AR as well. At least in our in vitro systems, when we use antagonists of ER or we modulate ER signaling, we haven't seen TMPRSS2 changes in the lung lineage cell lines and so forth. So
and no sex differences here, if you look at males and females, either in the mouse models or the human.
Right.
Thank you, Pete. Dr. Alamante, are you back? Andrea, would you like to pick up where you left off? Thank you.
So I was saying that the only way to convince a clinical colleague to run a clinical study was based on some data on patients affected by prostate cancer in androgen deprivation therapy. And for this reason, we look into this population in Veneto. So Veneto is a region of Italy that was strongly affected by this infection. And we look into in all the percentage of patients affected by prostate cancer in ADT therapy, either first or second generation ADT or in non-ADT. So this has been possible thanks to the fact that in Veneto there was the population of Veneto was very well studied. There was a very active initiative to collect all the clinical data of COVID-19 positive subject. This was crossed with other information of the Tumor Register Archive and the Regional Medicine Technical Commission. So the first thing that we notice in this population is that a male and a female have an equal prevalence of SARS-CoV-2 infection. However, male, in agreement with other data, have developed a more severe symptom when infected by SARS-CoV-2. This is represented here in terms of percentage of hospitalization, either in the ICU or by the rate of mortality. So when we look into the population of cancer patients, I'm talking about only male cancer patients, and we look at and we compare it with the risk of infection of the total male population. We observed that there was an increased risk of developing SARS-CoV-2 in patients affected by any type of cancer. This was associated also to a more severe syndrome. Indeed, there were an increased percentage of hospitalization in ICU and almost the double of mortality in a patient affected by cancer. This was equally observed also in a patient affected specifically by prostate cancer. However, the surprise came when we looked into the population of prostate cancer patients in androgen deprivation therapy. We compared this with the population of prostate cancer patients affected by SARS-CoV non in ADT therapy. So what we observe is that this population of prostate cancer patients in ADT have 4 times less the risk to develop an infection and 5 times when compared to the population of male patient affected also by cancer. These patients also, I mean, there are very few, of course. These ones that have been treated with ATP that also develop an infection didn't show like a severe syndrome. So to recapitulate, these are like really data that have been collected in a month. What I think they may be relevant because they show that cancer patients have an increased risk of SARS-CoV-2 infection. The prostate cancer patient receiving ADT therapy have appear to be protected from the infection, so have a better outcome. And this maybe could inspire other colleagues to run a clinical trial. And at the moment, we are thinking about to different way to study this by also complementing the study with some laboratory analysis. We believe that it's really possible that transient androgen deprivation therapy in patients with have, for instance, comorbidity could decrease the risk of the patient of hospitalization or of ICU or non-ICU. And what we are interested also to study is to understand whether there could be an effect of this androgen deprivation therapy on the reduction of the viral load or time to negativization in a patient affected by this terrible disease. So this is my last slide.
Thank you, Andrea. Andrea Miyahira, could we have the screen back? There we go. Thanks to all the speakers. There were a number of questions in the chat room as well as in the Q&A area. I would like, and I don't have everybody's cell phone number, so I can't adequately communicate with you privately, but if Peter Gann and Lorelei Mucci are present, and if Andrea Miyahira could bring you forward, I know you both had questions for Dr. Alamonte, and it would be greatly appreciated if you could do that.
If you could raise your hands, I can find you on the attendee list. And Dr. Alamonte, you can stop sharing your screen.
Hi, Andrea.
Hello.
Hi, Peter. I
Yeah,
know both of you,
I go first.
I know both of you had questions. Lorelei is still muted, so Peter, why don't you go first?
Sure.
Introduce yourself.
Yeah, so I'm Peter Gann, and I don't normally study infectious diseases, but I came across the Hoffman paper in March and was kind of blown away when I discovered that TMPRSS2 was involved. And I was very excited to see Dr. Alamonte's study, which was done very rapidly using administrative data. And I think it raises some very, very important questions about how we might do some further epidemiologic research to clarify this association. Obviously, the thing that we're concerned about in this kind of study is common to most pharmacoepidemiology studies. And it goes by the term confounding by indication. So the problem is that men receiving ADT for prostate cancer differ not only from all men, but certainly from other men who have prostate cancer because they have been prescribed ADT. And this means that there are a lot of potential confounders. And I think here, the one that raises the most concern is a huge confounder could be obviously the opportunity to become infected. And so, for example, if men on ADT are more frail, are they more likely to be sheltered and therefore A, less likely to encounter the virus, or B, more likely to be tested if they do because of their vulnerability. And this is not an easy thing to handle, but I just think that we need to look at it. The other thing that I would suggest is that we look at other cohorts who are on ADT, such as men who are or having androgen dysregulation pharmacologically, including transgender patients, as well as testosterone replacement therapy, which is fairly widespread. And not least of which is 5-ARI exposure, which is extremely common. So I'll let Lorelei comment.
Yeah, no, thanks, Peter, and thank you actually for just a really wonderful presentation. Also, I was wondering if it might be possible to pull up the flow chart that showed the study population in Veneto. One of the questions I had sort of relates to, and I think it's along the lines of the questions that Peter was raising, which is in the 2 million plus, yeah, exactly right here. So you can see nicely the number, the 4,500 individuals who are positive for SARS-CoV-2. And then you have the remaining more than 2 million men who are negative. So I guess my one question is, were they actually all 2 million men tested and therefore we truly know they're negative, or is this also sort of a diagnostic bias so that, you know, and I think you raised this in your paper, that men with prostate cancer, in particularly those on ADT, may just have been less likely to have gotten tested, and that's why there appears to be this inverse association. So I guess that's one comment I would love to hear an answer to. And then the second comment, I don't know if we can go maybe to the next slide, because I think this data was very interesting, looking at next slide after that, and slide 7 it looks like. I'm sorry, could you go to the next slide after that? it was interesting, yeah, here looking at, so, you know, and again, and I think you were cautious in interpreting the data based on the fact that it was 4 total cases. And I think what was interesting to look at is if you look at just the incidence of severe disease in the prostate cancer patients on ADT, the prostate cancer patients not ADT, and then individuals with other cancers, actually the prevalence of severe COVID infection is about 25% in the men on ADT, about 27% in prostate cancer patients not on ADT, and actually lower in men of other cancer patients. So, you know, it's interesting to, when we're thinking about ADT, is it about prevention of prevention of COVID infection from happening, or disease severity? And I think these data would suggest, even though numbers are very small, we might be cautious that it might not lower the incidence of disease severity. So I'd love to hear comments on both of those questions.
Well, again, I'm a cancer biologist and oncologist, so I'm not an epidemiologist. So I was very, I discussed almost 2 days with all the epidemiologists to try to interpret this data, right? And I agree with you that, there could be a population bias. However, you should be aware that the population of Veneto was very well studied in terms of both serological study and has been one, apparently, I mean, in Italy, again, has been one of the most characterized. So, of course, there could be this bias that not all the 2 million of men have been tested, right? But has been extensively tested between the Italian region. And the other bias that, I mean, the number, of course, are small, right? I try to ask around if other colleagues could also observe something similar. And for instance, I can tell you, I mean, that there is a small study also done in Milan with the last cases, which confirm also this observation, right? And again, they could be another bias that could be the social distancing. So it could be that the patients that are affected by prostate cancer that in therapy with androgen deprivation therapy do not need to go to the hospital for their treatment. So they stay more often at home, right? And this could have introduced a bias. However, this study was done in a moment in which there was a lockdown of the country in Italy. So this also should be taken in consideration. So that, did I answer correct to your question? I mean,
yes, yeah, I think that was helpful clarity. I don't know, Peter, if your question was answered regarding the issue of confounding by indication.
Well, I just think I mean, you know, administrative data sets can be analyzed very quickly. And I think that's, you know, that's a real strength, but I think often they have very limited opportunity to look at potential confounders. And I'm curious to know though, because there was a cancer registry involved as well as a drug database. And in the drug database, it sounds like you were not able to ascertain whether their ADT use was current or past at the time of ascertainment, or at the time of infection.
So we are currently trying to look into this data, right? And at the moment in which we send this publication, that originally was a letter. So never happened to me that we send a small letter, right, to Annals of Oncology, and the editor called me. They like, do you want to do like a paper, right? So so it went really very fast. So we are still analyzing this data, and more importantly, I mean, this prostate cancer COVID-19 register is still ongoing. So yeah. I'm trying to continue to do research with this database.
I would suggest if you have access to data on the actual indication for ADT, that would also be great. So is it possibly for men who are not candidates for surgery, possibly because they have high comorbidity or highly metastatic disease, versus men who've just gotten biochemical recurrence and actually are not all that unhealthy. And then you have the men with castrate-resistant disease, and that, that's a whole other kind of sub-cohort. So I think all of these questions, if we could get answers to them, and if there's a way for you to dig further in the data, would be fantastic.
Thank you, Peter, and thank you, Andrea. We also have a few other questions pending. One from Larry True at the University of Washington. Dr. True, if you could take yourself off of mute.
There we go.
There you go.
Okay. Thank you, Howard. Yeah, so as soon as TMPRSS2, I became aware of it, and the literature reminded me of the studies that I'd done with Pete Nelson and Jared Lucas in Pete's lab, who had developed the monoclonal antibody to TMPRSS2. And so what I wondered specifically with, I guess, Arul and Carlos is if you had looked at expression levels of the protein in men of a similar age controlled for other variables that lead to differential infectivity and disease progression, to see if expression levels of TMPRSS2 or of ACE2 and androgen also could provide a basis for understanding that interpatient heterogeneity in infection and disease. And Arul, I would also be interested in just learning which antibody you used, as I've begun reworking up the antibody that we had used.
So I can answer maybe part of the first part of the question, which is that data is being produced as speaking. So some of the data that I show you with the differential staining of some of the phenotypes on the androgen receptor antibody, TRPS2 and ACE2 are being done in the autopsy series that we have, as well as in a group of tissues from the pre-COVID area to have true negatives to compare with what we are going to be describing. And maybe, Arul, you can also give your thoughts and also your preferred antibody.
Right. I would say that we have done most of these studies using in situ probes for the most part, looking at the transcript. Primarily because of issues of getting the antibodies to work and so forth, although we have a nice antibody working for, of course, AR, ACE2, and most recently TMPRSS2. I'd have to get back to you in terms of which exact antibody that was, but I do know that we probably purchased a large percentage, or possibly all of the commercially available TMPRSS2 antibodies, and I think one might be working by immunohistochemistry. But I had thought that, Larry, that you might have Pete's original TMPRSS2 antibody to work with. I don't know if that was a polyclonal or what that was.
Exactly, yeah, and it's a monoclonal. I can, if I can share the screen, I think I could show you, yes. Just an example of that original study where we showed differential expression. Cancer and prostate cancer primary and cancer of different grades. And after working on the antibody quite a while, we got a very clean signal exemplified here. So yeah, I'd certainly be interested in sharing the antibody information with you.
Great.
Thank you, Larry.
Thanks, Howard.
We have Chuck Ryan in the queue. Chuck had a question on TMPRSS2 expression.
Yeah, so the interesting finding that TMPRSS2 expression is AR driven in the lungs is interesting. However, it cannot be the only regulator of TMPRSS2 expression. Are there other steroid receptors that drive this expression? And in particular, the GR, for example, would that be relevant? And patients getting steroids and things like that. And how do we tie all that into sort of standard physiology in the lungs and what the androgen receptor might be trying to do?
That's that's a great question. In our cell line models, we've looked at regulators of ER and GR, and we have not seen regulation of TMPRSS2 in that context. We've even screened other possibly more, I would say, non-specific inhibitors of transcription, like even BET inhibitors and so forth, or epigenetic regulators, and we still have not identified something that's consistent in terms of regulating TMPRSS2 in the lung lineage. But certainly that was sort of our thought, that AR is possibly not as consistently expressed in the lung as possibly GR and ER, that they would potentially be able to regulate TMPRSS2 expression, at least in some subtypes of cells in bronchial or alveolar epithelia, but we don't have evidence of that. Certainly not in the murine models, but also in the lung cell lines that we've assessed.
Thanks.
Great, thanks.
Josh, this is Carlos. Just going back also to the question of Larry. I was able to get the antibody that we have been using for some of these studies, and it's a monoclonal antibody made in rabbit from Abcam, and it's clone 92323.
Okay, thanks, Carlos.
That's great.
And if there are a series of different antibodies, we might think moving forward of validating the different ones and comparing them?
Absolutely, Larry. I love the staining that you had, which is a little bit different from the one that we see. It seems like it's more secretory luminal, so that would be very interesting for us to study.
It is.
The other thing to mention is that it's nice to correlate the protein and antibody staining with, of course, the in situ transcript expression to make sure that the right cell types are indeed lighting up. Because it's certainly, in terms of in situ probes, you can get very specific in situ probes, and there's less issues of non-specificity when we're doing in situ type analyses for RNA.
So our last question will be to Dr. Loda. And before you ask your question, I would just like to comment that Julia Arnold from NCI kindly put a message up in the chat room about NIH supplements to current grants for COVID-19 research, which I thank you very much for doing that, Julia. This is a smart thing, and people need some extra support for all the work they've been doing outside of the aims on their grant proposals. Also, if anybody has ideas for other of these types of webinars that you would like to do, please send Andrea or myself a message, or preferably both of us, and let us know what you might be interested in, and maybe you'll lead one after we move beyond TMPRSS2 in the very near future. Okay, so Max Loda, good to see you.
Good to see you too.
What is your question?
Oh, I didn't have a question. I just tried to answer Pete Nelson's question about ER alpha and beta expression, and I just quickly looked up, and there seemed to be, at least from the images that I saw, type 2 pneumocyte, not pneumocystis, as was wrongly corrected, expression of both alpha and beta in the lung. So that's one thing that I wanted to say. I also wanted to reiterate what Carlos said about, or somebody said about the neutrophilia. We see a lot of neutrophilic pneumonitis, and we found, increased expression of neutrophil extracellular traps, which have been together with the Cold Spring Harbor group. So that's another area of pursuit in terms of therapy with DNA, intratracheal DNAs and things like that. So I just wanted to bring that up to the forefront. But I didn't have a specific question. One question I wrote earlier is, I think that our rule showed that there's co-expression of ACE2 and TMPRSS in the prostate, albeit small, and I wanted to know which cells those are, but I don't know if you identified those or not in the normal prostate.
I think that was probably Carlos, was it? I don't think that I showed much data on prostate. No, it was all lung.
Never mind then. But thank you. This was fantastic.
Yeah, sorry. From Doug Strand's data set, it was in the Sugnac paper. They showed it was in Hillock cells.
Ah, sure.
I see.
Right. Max, you were saying?
That's it.
Okay, thank you. I am overwhelmed by the enthusiastic attendance at this conference. Thank you to all the speakers, my co-conspirator, Andrea Miyahira. And please, if you have burning ideas that you would like to be seen addressed in COVID-19, outside of COVID-19, our goal is to keep our community engaged and supported throughout this crazy period of time, which hopefully has an endpoint sooner rather than later. But thank you all, and we say stay safe and let us hear from you. Thank you.
Thank you.
Thank you.
Thanks,
Thanks, everyone.
Thank you very much.